Hi guys, in this video we're going to be looking at how to extract DNA from living cells. For this you're going to need some sort of vessel to contain the DNA in. I've got a test tube in this case. You're going to need some sort of washing up liquid, some salt and some form of alcohol. I'm using isopropyl alcohol, but some sort of strong alcohol that you can buy in the shops off a shelf is absolutely fine. Okay, so what we need to do here first of all is we need to obtain a sample. You can use saliva for this. I know it's not the most pleasant prospect, but it's something that's readily available that your whole class can contribute their own sample to. And also, I suppose there's some sort of I don't know, it lends it some authenticity when you're actually, when kids are getting the opportunity to see their own DNA. But if that crosses a personal line for anyone, you can easily extract DNA from any other living cells. A piece of fruit, an onion, that sort of thing. They're commonly used. But first of all, you need to get your DNA sample. The next thing we need to do is we need to destroy the cell membrane. So the DNA is neatly packaged up inside the cell. In fact, it's not only within the cell, but it's within the nucleus within the cell. So it's basically double bagged, if you like. We need to burst the bags that they're in. And washing up liquid is a good way of doing this. So we're just going to add a little bit of washing up liquid. And what this does is it degrades the cell membrane. Now, if you were using a piece of fruit or something for this, you would probably want to actually mash up the fruit, just use something to pulverize it a little bit. But in terms of just using saliva or something like that, then just adding the washing up liquid and mixing it up thoroughly is fine. So that should have now burst the cell membranes, and the DNA is now floating free within there, but we can't see it. We can't see it. What we need to do now is we need to find some way of actually gathering that DNA together in one place. So we're going to use the salt, put in a little bit of salt, and this is just to kind of, I suppose, kind of like coagulate it. Oh, a bit too much there. So we've added in a wee bit of salt there, and what that's basically going to do is it's going to allow the DNA to sort of stick to it a little bit. Okay, so you can kind of see white strands starting to form, and that's basically the salt sticking to the DNA, but not highly visible at the moment. It's kind of working. I'm seeing it there. If I took it very close to the camera, you'd be able to see it. But what we need to do now is we basically need to stretch out the strands. Okay, so we've stuck it to a bunch of salt that makes it visible, but now we're going to use a very light substance, in this case alcohol, whose density is somewhat lower, and it's going to now draw out the strands of DNA. So that's the only purpose of it. This isn't involved in any sort of reaction. It's just to stretch out the strands and make them more visible. Now you can pour it in directly. I'd actually given myself a pipette there, and if I was doing it for a whole class, I'd be using a pipette and giving them a set measure. But just because this is my own version of it here, it should be fine. And what we can see, and actually this has worked quite well, I'll take it up to the camera, and you should be able to see we've got this the washing up liquid at the bottom there. Okay, and you can see sort of white filaments running through it and all the way up up the column there, and that is DNA. It's been coagulated by the salt and then drawn out by the lower density of the alcohol there. So you can get your whole class doing that, and they find it quite a thrilling experience, and it teaches them a little bit about the fact that, well, actually DNA is in pretty much all of your cells, some exceptions. And so it gives them the opportunity to extract their own DNA. It's not the kind of thing where you actually get to see the double helical structure of it or anything, but it gives it some authenticity, some realness for the kids. Plus, it's an experiment that you can then write up as well. So you could use the experimental methodology, sort of using aim, hypothesis, materials, method, results, conclusion, laying it all out like that. Okay, cheers.